A genetically modified organism, or GMO, is an organism whose genetic material has been modified, usually by a form of genetic engineering. Genetic engineering is the technological process of altering the genetic material of a cell to make it capable of performing desired functions, such as producing novel substances. A GMO is also referred to as a biotech crop or transgenic, meaning that it has genes from another organism that have been put into its genome through recombinant DNA genetic engineering techniques. Genetic engineering of crops is becoming a prevalent form of crop improvement. Genetic engineers and biotechnologists alter the genome of crops by inserting the gene containing a desired trait into a crop to induce the expression of this trait, which would have not normally been a part of that crop's genome. This gene is normally found naturally produced in a line of the same crop or a different crop genome, and introduced into another line of a crop that already possesses other favorable traits in order to increase its yield and improve its yield reliability, or enhance its taste or aesthetic appeal, or to induce or improve its ability to withstand unfavorable weather conditions, such as being freeze resistant. Most of the genetically engineered crops grown today are altered to be resistant to herbicides, so that the farmer can spray herbicides to kill weeds and their valuable crops will not be adversely affected, or the crop is altered to produce insecticides that kill insect pests that would otherwise kill the plant or cause the farmer to have to spray insecticides on the crops. Some crops are engineered to tolerate or repel fungal and bacterial pathogens. The most common method of inserting a desired gene into a crop is by microbial vector. Agrobacterium tumefaciens is a naturally occurring soil microbe best known for causing crown gall disease on susceptible plants. It is an unusual pathogen because when it infects a host, it transfers a portion of its own DNA into the plant cell. The transferred DNA is stably integrated into the plant DNA, and the plant then reads and expresses the transferred genes as if they were its own. By substituting the DNA of interest for the crown gall disease-causing DNA, scientists derived new strains of agrobacterium that deliver and stably integrate specific new genetic material into the cells of target plant species. Another method is electroporation. In electroporation, plant protoplasts take up macromolecules from their surrounding fluid, facilitated by an electrical impulse. Cells growing in a culture medium are stripped of their protective walls, resulting in protoplasts. Supplying known DNA to the protoplast culture medium and then applying the electrical pulse temporarily destabilizes the cell membrane, allowing the DNA to enter the cell. Another commonly used technique is the gene gun method.
Major alternative to the use of agrobacterium to get genes into living plant cells is a physical method called the gene gun, which literally shoots gold or tungsten particles into living plant cells, carrying DNA on their surface. The first embodiment of this by the fellow at Cornell, John Sanford, who invented it, actually used a .22 caliber blank to accelerate a plastic bullet with a drop of tungsten and DNA on the tip towards plant cells. The plastic bullet would hit a donut-shaped plate with a hole in the middle, and the tungsten would spray out the other end, but the plastic bullet would stop, and the plant cells in the bottom would be bombarded at gunshot velocities. It turned out it worked. Once the tungsten particle goes into the cell, the DNA would simply fall off of it and once in a while find its way into the nucleus, and by just random DNA integration find its way into the genome.
Environmentalists have three main concerns. The GM crops will create superbugs and superweeds or reduce biodiversity, but above all, they're worried about the irreversibility of GM crop releases. In the open environment, pollen from transgenic crops could pollinate another related plant. If they cross with their wild relatives, there could be the danger of creating superweeds resistant to herbicides. And if they cross with non-GM crops on organic farms, for example, the farmers couldn't guarantee the non-GM origin of their food product.
Biotech crop hectares increased 100-fold from 1.7 million hectares in 1996 to over 170 million hectares in 2012. This makes biotech crops the fastest adopted crop technology in recent times. In 2012, developing countries grew 52% of global biotech crops, and industrial countries grew 48%. The growth rate for biotech crops was at least 3 times as fast and 5 times as large in developing countries at 11% versus 3% in industrial countries.